Okay, let me see. Well, first of all, thank you, Greg, for inviting me. I have to admit that I'm not a software engineer, so today I just want to kind of talk from a user's perspective, um, how we can make sense of the multifish pipeline with Napari. I guess some of the rationales why there is so many tools out there, and what we use for which application, and also I was hoping like to get more inputs from you guys on how we could further improve and maybe create a more user-friendly interface for visualization. So for people who are not familiar, I worked with a multifish project team, and we developed a pipeline, or we developed an experimental setup where we were able to image 6 tissue volumes for RNA fish. So we were able to detect transcripts or gene expression in cells in a sick tissue. So in order to do this, we use combined expansion microscopy, and we use light sheet microscope. And the images we get are these images where we have a cytosol stain that we use for segmentation, and then these pantas that will have to extract the spots so that we can count how many transcripts are in each cell. And in order to do multiplexing or to probe for dozens of genes, we image the same piece of sample like multiple times. So in most of the experiments, we do it for like 10 image rounds. So we have 10 such big chunks of data that is usually like 10 terabytes in size. So then the next step we normally do is we stitch this data after they are collected on the light sheets, and then we register these images across different rounds, and we segment based on the cytosolic stain, and we perform spot extraction, and then we will get basically a spreadsheet that tells us where the cell is and what kind of gene is expressed. So this is really largely a collaboration, within Janelia, as well as with people at the Max Planck, and really many of the software engineers have helped out, and especially with building a very convenient pipeline. So I am really coming from like experiment background, molecular biology background, I would say. So I guess just trying to think from a perspective how we can make use of the outputs of things that come out of the pipeline, and make sense of the data. So just a brief overview of the data format. So this is what we have. We have basically this pipeline output is you can have if you have 2 rounds of images, you have 2 folders. If you have more than 2 rounds of images, you have more than 2 folders. Basically, the number of folders equals the number of image rounds you have. And within these folders, and also specifically, this pipeline was developed by Conrad and Christian in software engineer, in SciComm. And in these folders, there is multiple subfolders, and we have the stitching where there is an N5 file that contains the stitched image volume. And then we also have and we also have a spots folder where you have the extracted spots in a TXT file. We also have a segmentation mask. It's a TIFF file. We also have the content spots. Basically, these are like CSVs where the columns are genes names and the rows are cell IDs. And then in a different round, and most of the folders are the same, but in this case, you will have a different folder called registration. This is the image that has been registered and is placed here. So this is a summary of these image format. So we have image data that's in N5 or TIFF format. We also have spots detected in the CSV format. We also have these cell ID or their positions, these kind of metadata. It's also in a CSV format. So a question for us is, how could we make use of this data for data visualization with specific goals, such that we can understand how our image acquisition goes, whether there is any stage movement that has made the image acquisition fail, or if there is any, did the pipeline work, did our registration work, did the stitching work, did the spot detection detect spots properly, and did the segmentation segment cells properly. Also, lastly, we also want to know whether the data makes sense, whether we detect the genes in the areas that we're supposed to detect, where these are supposed to be expressed, and whether there is any false positive or negative detection. And I also want to say that we also use a lot of other tools, such as, I guess I have to say that we are heavily dependent on the Fiji community, where we use the N5 viewers many times, a lot of the times, and also big data viewer. And also most recently, we are also using the Stefan RS Fish, where they have this big data viewer based spot inspection. They are all very helpful. And we also use the Pantera for segmentation, manual annotations for training the segmentation pipeline, and we also use the track EM2. But okay. So, but why do we want to use Napari at the same time? Because I think there is two main reasons. One reason is that we want to see segmentation, raw image, and the point cloud at the same time. And I mean, at the time when I was trying to visualize this data, I do not know anything that I could use out of the box that would allow me to do this. So that's why I chose to use Napari, and also for other reasons, such as I can do my plotting and some statistical analysis or other data analysis in the same kind of ecosystem. So in the Napari, I'm mainly using the 3 different layers: the image layer, where I load my raw image data, and then the labels layer, where I load the segmentation masks, and also the point layer, where I load more point cloud. And this is just my code. I have to apologize that I have some issues with my VPN, so I can't show the Napari in real time, but this is kind of the code I use is really simple, where I kind of load the TIFF just by reading the images in, and we can also load the N5 files using ZAR, and then we can add them to Napari for viewing, and then we can also load the CSV files and add them as points, as mentioned. And also, I guess I was also at the time working from home. I was also able to just crop and look at a very subset of data, and I can look at that on my laptop, which was super nice. So for segmentation, this is a TIFF file, so we kind of have to crop it. But if it is a N5 format, then we can just load a subset of the data. And also, I think Napari support task arrays, so we can also load these ZAR files as task arrays, and then access the subset that we're interested in. And also, we can just filter out the spots that in this particular crop, and then we can, this way would allow us to look at all the modalities at the same time. So we can evaluate how our segmentation works, and also how does our spot extraction works. And also on top of that, how does the registration work?
Can I ask a few questions at this point, Yuhan?
Yes.
My first question is just in the, when you create the Napari viewer, I haven't used Napari and I'm very interested in using it. So I have some really basic questions about it. What is n display equals 3?
I think it's a dimension. So yeah, I think that it's a dimension. This is a 3D display.
Okay, cool. And then not in the notebook, but on the examples of like how you crop and size your data to be loaded into Napari, the lines at the bottom, b equals spot.copy. At that point, is spot a pandas data frame?
Yeah.
And therefore b is also pandas data frame, just a copy?
Yeah.
Okay. And that's because I don't use those that often. So that's pandas data frame syntax for filtering based on range for a particular like row X,
Yeah.
Y, and Z. Okay, cool.
Yeah, but I guess you can also just load as you know, the NumPy array, and then use the logical basically a kind of like crop that way.
Yeah, I don't think one's better than the other. I just want to know what the syntax is and how it works.
Yeah. And then I guess another big reason that I thought this was super handy was because sometimes we want to search for a particular cell with their ID or a fish spot with their spot ID. This is kind of like scenarios where we're doing a lot of inspections. So we want to know how the segmentation works or how the spot detection works. In this case, when we saw some big cells where the cell size is very big, we want to know, are they merges? In our case, we have millions of cells, so it's kind of difficult to like look at an image and look for a particular cell that way. So this really helps us to kind of query a particular cell with their ID. Or if we have adjacent cells that have very highly similar gene expression patterns, we also want to know if they are over-segmented, if they're just a same cell, but they get split into two segments. And also for spot inspection, we also want to know like cells that have very high intensity values. Are they a blob of multiple spots, but they're just too close and they were detected as one big spot? Or we want to know with the spot that it has very low intensity values, are they background? For example, in our case, there are some lipofusion signal, or there could be other background signals that get detected. So these are kind of the things that we do to evaluate how well the pipeline has worked for a particular data set. And then additionally, we also later on, when we actually do the real data analysis, we want to know whether this data makes sense, right? There is a lot of biological confirmations we want to do. And I will give an example of that later. Yeah.
Yuhan, just a quick question. For these analyses, how interactive is it? Do you need to be there inspecting it, or is there any part of this that could be automated? Like you could just take this code and put it into the pipeline?
Well, yeah, that's a good question. This right now, this is kind of like pieces. I would love to have this as part of the pipeline. I think this is probably something, if hopefully we would be able to have a chance to work on. It's not too difficult. But this is kind of, I would say, semi-automated. For example, the segmentation inspections, we have some code where we can flag cells that we think are likely over-segmented, because these cells, because we've kind of prone our algorithms to over-segment rather than under-segment because they are easier to correct. Then we have some code where, based on their gene expression similarities, as well as their distances, whether they have overlapping or neighboring pixels, we kind of flag a subset of these ROIs, and then we can just search for their cell ID, and then we evaluate whether they are true oversplits or if they are just two cells that are very close together. So that's something that we have. So that flagging part is automated. The search part can be automated, but then the inspection part is manual.
Right.
Yeah, and then the same goes with spot inspection. So we can plot the histogram of the spot intensities, and we can pick a certain cutoff based on some automated thresholding algorithms, and then we can evaluate whether these are proper threshold. Yeah. So, and then this is just a snapshot of the code I use, where basically I can just, this is kind of very simple, but I just kind of type in my cell ID that came out of these, these analyses. And then based on the cell's position, I can crop out a subset around this cell. So I know what is going on in that particular crop. I can look at the segmentation, the raw images, and also spots detected in this particular cell and its neighbors. And also just, I guess, I want to give a also interesting example, I guess, was that I can also combine this with, for example, interactive plotting, since in Python, like Plotly, where I can plot the expression of, this is Vgroup 2, which was supposed to be expressed in excitatory neurons, and Vgat, which was supposed to be expressed in inhibitory neurons, and ask, this is kind of trying to evaluate whether our experimental method actually works well, because you would expect there's no Vgat spot detected in the excitatory neurons and no Vgroup 2 spot detected in the Vgat neurons. or in the inhibitory neurons. But then we saw this group of neurons that have expressed both VGRU and VGAT. So this makes us wonder whether we have some false detection or what's going on in this area. And because Plotly allows this interactive selection, where you basically can identify the cell IDs this way. So we would be able to pull out the cell IDs this way. But of course, you can also just kind of grab everything within this area and kind of search for all the cells in this area and plot or display all these cells and look at what happened. But in this case, if we just look at a few of these cells, use the method that I just mentioned, we can look at what is happening here. But actually, interestingly, we found that in fact, there are a group of neurons that co-express these two transporters, and there are recent publications suggest that there is co-packaging of opposing neurotransmitters in individual synaptic vesicles in this particular area. So I thought, this is pretty interesting. Really, it kind of helped us to kind of validate our data and also enables, more novel scientific discoveries.
We have seen the same thing in the whole brain zebrafish, by the way. And because we looked at the same comparison, excitatory inhibitory, with the expectation that they'd be mutually exclusive, and to the large extent they are. And in at least one case, this test did reveal a problem with the sample preparation, where a harsh stripping of the probes actually caused some fragmentation of the spots, and they became more widely distributed throughout the brain. And that mixture of these two cell types was artifact, right? Mm-hmm. But even in our best prepared samples, we see a similar type of population with a little bit of mixing, and there's single cell RNA-seq data in zebrafish brain to support it as well.
Yeah, that's interesting to hear. But yeah, so I guess you can also imagine that instead of plotting a scatter plot with two gene expression, we can also replace this with a PCA or UMAP, displays all the cells, the molecular cell types. And then the same goes that you can select for subsets of population and display the subset of population and see whether they are spatially enriched or if they are kind of evenly distributed across the tissue volume. So I think that would be quite interesting to have a Napari-based plugin for that. But I guess that's why I have proposed future how-to topics. And I'm still learning a lot of these, but hopefully kind of invitations for some future discussions on these topics too. So some of them, first, so how to run Napari on the LSF cluster. I'm not sure if it's possible. I haven't talked with people in SciComm, but based on my experience with some of the people around the lab, it didn't sound like anyone has got this running yet. And also how to run Napari for multi-scale visualization, because I know that Napari could run an image pyramid, but I am not sure if the N5 can be easily converted to the image pyramid and run with Napari, because that would be quite nice so that we can see the images at different scale rather than what I'm doing, kind of like cropping the image. And also how to plot in Napari console. So if we can do what I just did with the Plotly in the console and select the cells there, that could be also quite nice. And also, I think this is there is some threads on making videos with making beautiful videos with Napari, but my experience with this was it's pretty slow for me, and so it would be nice to learn more on how we could make videos with Napari for presentations. And lastly, whether we could or how can we create a Napari plugin in this case? Can we combine some of these plotting functionalities and so we can basically visualize the dimensionality reduction and see where the molecular clusters are, and then also to see where they are in space. So that could be very interesting. So yeah, so that's all I have.
About the second point there on your wish list, at least the stitching software does output an image pyramid, right? So you at least have one data set that's where the resampling levels exist to test with. The resampled registered data doesn't bother to create an image pyramid. I think it might only create, it might create more than one level, but it's not like a full pyramid.
Oh,
But it could certainly, an extension could be written to take like an N5 input that has one scale level and create all the same scale levels that are in the stitched output. It's just you have to pay for the resampling, right? Which makes sense if you want to visualize that result. But previously, the goal was just put the data in the same place. And then if you want to view it interactively, we probably should make those pyramids.
yeah, I think I would even so I'm even having troubles just looking at the stitched what you were talking about. So the stitched files with that is the N5 format. I guess I probably should do more reading and study that more because I had some troubles just loading that in as a pyramid because what I'm currently doing is kind of like loading it at different scale. And as a one NumPy array or a Dask array. But I'm not sure. I haven't successfully loaded it as a pyramid. Basically, just having the all the images with all scales. So yeah, so that could be interesting to learn how we could do that.
This is a great wish list though. And I think I'm going to ask, future presenters to always have a slide like this at the end. Like, what would you like to see as how-to presentations that are like related to what you talked about? It's really helpful.
That's great. Yeah, we look forward to more on this.
Yeah, I mean, I'm not sure, but maybe we can get Nick to come answer some of these questions or anyone else who's, gotten to use Napari a lot.
Yeah, that would be awesome.
I think maybe we have a question from Daniel who says his microphone is not working. He says he's also interested in Napari for drawing ROIs. Yeah, I suppose I'd be interested in that too. I'm not sure if you've tried any other tools for that, Daniel, but Yuhan mentioned one, Paintera. I also use a desktop client for that sometimes called ITK Snap. There are a couple other Fiji plugins for it too. They all have strengths and weaknesses, and I would love to do it with Napari, especially because you could memory share with processing in Python, right? Like draw your segment and then run some SciPy processing with your code in your notebook, reload the segment in Napari, and it's all very like seamless. It would replace a lot of manual kind of shuffling things around with files. Let's see, built my own PyQt graph client, but would like to try with Napari. Oh, okay, so you made your own Python-based ROI editor, but yeah, it'd be, there's got to be. Have you done any manual work in Napari?
But primarily because you can definitely annotate in Napari, but I don't know about the functionalities. I don't think it can interpolate across different, because we have these 3D images, the cell are in 3D. I don't think it currently have these interpolations in Napari. Might be able to do it elsewhere, but that's something that's why I didn't do it there. But that's why I kind of tried Pantera and, as you said, Pantera and TrackEM too, these kind of applications.
Another potential solution would be to have your primary workspace be a Jupyter notebook where you can do visualizations in Napari and manual editing in another Python-based tool like what you built in PyQt Graph, and just make sure that they can handshake data with each other. So if you make an edit in PyQt Graph, you'd rerun a single Python function that reloads the data quickly in Napari for, maybe now you have multi-layer visualization and you have arbitrary slicing and things like that. So there's maybe just, maybe the solution isn't forcing Napari to do the thing you want, but just getting it to communicate with the thing that already does what you want. Any other questions before we move on to talking about compiling Julia code?
No, I just comment. I think some of those things are in the works. I know Davis is working on SAR multi-scale specification, and I think there is a multi-scale option that was recently added, but it's kind of a Boolean. You have to manually turn it on and off, and manually feed in like an array of scales. But I think hopefully when Davis gets done with that, it should be automatic.
Yeah, that's great.
He just presented, I think, a week ago in the code review group, his code for doing multi-scale construction of multi-scale pyramids from a single input image. I don't think that was recorded, but maybe we can get him to talk about it in how-to as well, and kind of combine our thoughts about what a good multi-scale specification would be. Because I think it just comes down to formatting, right? All multi-scale images have the same thing in it, a bunch of resampling levels that are smooth, but how does the metadata look, and how are they chunked, and how do you access it? And all of those sorts of things are totally arbitrary, and every tool probably does it in a different way.
Yeah, it would be nice if it doesn't have to like create another copy of the data. or load everything, even a scale 0 into memory. Because I mean, at least for us, the Napari does not run on the cluster, so that could be a problem because this data with scale 0 is like the high scale or the highest resolution is pretty big. It's like 200 gigabytes. Yeah.
I think ideally though, you would want to run Napari, I think, on your own computer, but maybe the data is coming off the cluster.
Exactly. Yes.
But you still have to load everything that gets displayed on your screen. If Napari is running locally, has to be in memory on the local machine at some point, right? So, but I don't know how it works internally, how much it caches, you know, like if it tries to load the whole scale 0, then it won't work. But if it only loads the plane you currently need and everything else is on the cluster, then are you still on the file share? That's fine, right?